Well, hello, everybody. Good morning, good evening, good afternoon. Welcome to the 23rd session of the GO2NE Global Ocean Oxygen Network webinar series. I am Dimitri Gutierrez from the Instituto del Mar de Perú, IMARPE in Peru, and the whole GO2NE team is very happy that you took the time to be with us and our incredible speakers today. Before starting, we would like to bring your attention to the Ocean Oxygen website, where you can find past webinars and good news. There you have it on the screen. The good news are the Ocean Deoxygenation newsletter. The QR code will take you straight there. If you are on Twitter, you can also follow at good_ocean_oxy for the latest news and activities of the GO2NE community. Indeed, the next GO2NE webinar in November, so please look out for details to register when the webinar is announced. Some quick housekeeping announcements before welcoming our speakers. You, as attendees, have the possibility to type your questions in the questions and answers box in Zoom. The chat can be used to share additional information or if you have any logistical problems. Okay, saying this, let's get started. We have two great scientists speaking today. We will start with Montserrat Aldunate, who is based at the University of Concepción, Chile, followed by Michelle Graco, who is based at the Instituto del Mar in Peru. Both speakers have 20 minutes to present, followed by 10 minutes of questions and answers. And now let me introduce you Montserrat Aldunate. Montserrat is a marine biologist, Master of Science, and Doctor in Oceanography from the University of Concepción. In 2019, she joined the Coastal Ecosystems and Global Environmental Change Lab in connection with the Chilean Science Foundation project on carbon cycling and physiological traits, phytoplankton functional groups under low pH, low oxygen conditions. She developed her doctoral thesis at the Laboratory of Microbial Oceanography at the University of Concepción, the Instituto of Oceanography. In her thesis, she studied the assimilative metabolisms of carbon and nitrogen in pico cyanobacteria that inhabit anoxic marine areas in the North Pacific and the southeastern tropical ocean. Montserrat has experience in the study of marine microorganisms, phytoplankton, biogeochemical cycles, and the areas of oxygen minimum and low pH. She has developed experiments coupling stable isotopes, flow cytometry, and cell sorting. She has participated in several field campaigns aboard national and international scientific vessels, and in addition, she actively participates in activities and workshops for science dissemination among the community. So, saying this, Montserrat, you are welcome, and the floor is yours.
Thank you very much, Dimitri, for the presentation. Today, I'm going to talk about this work entitled Effect of the Oxygen Minimum Zone on Eukaryotic and Prokaryotic Phytoplankton on the Eastern Tropical North and South Pacific Ocean. This is a joint work with Jane Wong, Edson Piscoya, Peter Fondasso, and also Osvaldo Ulloa and Christian Vargas. Oh, okay. Phytoplankton plays a fundamental role in the marine ecosystem and biogeochemical cycles through the carbon fixation, oxygen production, and nutrient utilization. And phytoplankton participate also in the carbon export to the deepest waters. Its vertical distribution has a fundamental role or importance in the structure and dynamics of marine ecosystem, identifying light and nutrient as the main factors responsible for this vertical distribution. However, there are some places that present very particular phytoplankton vertical distribution. Those are the oxygen minimum zones. Most of you are probably very familiar with these anoxic or oxygen minimum zones. However, part of these oxygen minimum zones have been redefined as anoxic marine zones because they are mainly anoxic. Those anoxic marine zones are areas of the ocean located in the Eastern tropical North and South Pacific and also in the Arabian Sea. These zones present no oxygen in intermediate waters, are plenty of nutrients with a high nitrite abundance, and also have a high microbial abundance, and frequently we can find a secondary chlorophyll maximum that develops in the top of this anoxic layer that is mainly composed by Prochlorococcus, cyanobacteria, and in a lesser extent, Prochlorococcus and Synechococcus cyanobacteria. This secondary chlorophyll maximum develops below the canonical euphotic layer. That means is developed in very low light conditions. So the vertical distribution of phytoplankton in these anoxic marine zones can be split in three main layers. One, oxygenated surface layer that is composed by phytoplankton of different sizes with eukaryotic plus eukaryotic phytoplankton. Then a chlorophyll minima that is in the base of the oxycline, and a secondary chlorophyll maximum at the top of this anoxic layer that is mainly composed by this Prochlorococcus cyanobacteria. And these are special lineages of Prochlorococcus that has been only found in these anoxic layers. None of these lineages has been found in other places of the ocean. So, with these observations, we raise the following questions. One, what metabolic strategies use the phytoplankton that inhabits these anoxic marine zones? Do they photosynthesize? Do they use other strategies to produce organic matter in this low light environment? And why does a minimum of chlorophyll occurs when the oxygen concentration decreases? Do eukaryotic or surface phytoplankton need oxygen to survive? Now, I'm going to show you some of our work regarding the secondary chlorophyll maximum questions. Here is a work entitled Cryptic Oxygen Cycling in Anoxic Marine Zones. We here analyze samples from the secondary chlorophyll maximum. We took that samples and incubate them in different stations in the Eastern tropical North and South Pacific, and incubated using different light intensities. And we follow the oxygen concentrations. Here you can see in the panel B a net community production, that is the balance between oxygen production and consumption. And in the panel B, the gross community production based on oxygen production, that is net production plus respiration, and carbon fixation, that is the inorganic carbon incorporation into biomass. So you can see that this oxygen is increasing when increasing the light, the same for carbon, and they need very low light conditions to photosynthesize. Actually, this presents a plateau or a saturation of light between 10 and 20 micromoles per meter per second. So they need very low light conditions. However, in situ light is much lower. that is in the range of 2 or 5 macromolars per meter per second. So in this case, this is the range, oxygen do not accumulate. And that is why this is a cryptic oxygen cycle. Even this oxygen is low, is enough to stimulate important aerobic metabolism. You can see here the profile of fluorescence profile, and the primary and secondary chlorophyll maximum, and the oxygen profile. And you can see here a peak in the expression of the genes that code for enzymes related with aerobic respiration, like LATO and HATO, and also enzymes that are related with autotrophic nitrification or nitrite oxidation, oxidoreductase, sorry. And also an accumulation of trancrits that are belonging to Nitrospira, that is a nitrite oxidizing bacteria that peaks also with this secondary chlorophyll maximum, meaning that maybe they are using this oxygen to oxidize nitrite. So this low oxygen can be very important in stimulating these aerobic pathways in these otherwise anoxic zones. Other work we evaluate is the carbon assimilation, but the phytoplanktonic community inhabit this secondary chlorophyll maximum in these anoxic marine zones. And we took samples for natural abundance of delta carbon 13. That is the natural abundance of this isotope in the community of this secondary chlorophyll maximum in the Eastern Tropical South Pacific and the Eastern Tropical North Pacific. And also we took samples for natural abundance in the top of this anoxic layer when no secondary chlorophyll maximum was developed. And you can see that in the secondary chlorophyll maximums in the north and in the south, the natural abundance of this isotope was very variable, but there not present significant differences between the north and the south. But when we compares with the top of the anoxic layer when no secondary chlorophyll maximum was developed, we found significant differences being this community of the top of the anoxic layer very depleted in this carbon 13, and these values are similar to the values found in chemiautotrophic metabolisms as anammox and sulfur oxidation that we know are very are very active in these anoxic marine zones. We also make some incubation experiments, and we incubate samples of the secondary chlorophyll maximum community using bicarbonate to evaluate carbon fixation, using also glucose and acetate to evaluate organic carbon incorporation. And we found that when high abundance of Prochlorococcus, there are high abundance of Prochlorococcus, the photosynthesis exceeds chemototrophic chemosynthesis, carbon fixation in dark, that is the black columns. But the majority of the stations here, the carbon fixations or fixation rates were very low, and much carbon fixation was on average 5.7 times lower than the glucose assimilations, showing the prevalence of heterotrophic over autotrophic metabolism in the secondary chlorophyll maximum. Going back to the previous questions, and now regarding if autotrophic or surface phytoplankton needs oxygen to survive, we think that is a very good question, because phytoplankton are estimated to contribute approximately half of the global phototrophic primary production and are therefore a major contributors to oxygen on Earth. So why will oxygen affect them? In this work, we evaluate the relative influence of environmental factors that control the vertical distribution of phytoplankton and nanoplankton associated with these oxygen minimum zones. And the statistical analysis indicate that temperature, oxygen, and carbonate chemistry have a significant influence on the distribution of this pico and nanoplankton, phototrophic pico and nanoplankton. So what we did, we make some experiments using strains of Imantonia that were isolated in these same areas, but in the oxic layer. And we try to evaluate the combined effect of low pH and low oxygen that are the characteristics of this anoxic or oxic bond, that is a pH of 8.1 and oxygen in saturation, and in OMC conditions a pH of 7.5 and oxygen 55.6. And you can see here the pH in time, the oxygen in time, and the in vivo fluorescence. The controls show that they are an increase in the pH because of intense carbon fixation, high oxygen production, reaching saturation. That means that it's active photosynthetic metabolism, and also an increase in the fluorescence. That means that this strain is growing. However, when we put the same strain in OMC conditions, we saw that there were not changes in pH or oxygen. And if we see fluorescence, we saw that this strain do not survive. So oxygens can be affecting this strain. So with all this observation, we try to show you some some hypotheses that can be explaining this pattern. And what we do, we took or we try to relate the phytoplankton, the abundance of phytoplankton, oxygen, and light penetration through 147 published profiles from the Eastern Tropical North and South Pacific, using data from flow cytometry, also hydrographic data by means of CTDO and fluorescence, and we evaluate some possible hypotheses. One of the hypotheses was that grazing by protists, but there were not evidence of flagellates increase in chlorophyll minimum. That means that there are not evidence of grazers or a rising of grazers in this chlorophyll minimum that can be explaining this low this low fluorescence in the base of the oxygen. Other hypothesis was the irradiance, but fluorescence minima occurs at level greater than 1% of the incident light. You can see here that dots represent the minimum of chlorophyll in this that was in the base of the oxycline, and the majority were between 1 and 10% of the incident light. That means that have enough light to photosynthesize. In addition, there are a variety of phototrophs that inhabit very low light conditions. There are diatoms, coccolithophorids, cyanobacteria, etc. So the light alone does not limit the vertical distribution of phytoplankton in oxyclines. And other of the possible explanations is an inhibition by oxygen. You also can see here the dots represent the minimum of chlorophyll of these different oceanographic cruises and the oxygen concentration. And you can see that in average, the oxygen concentration for this chlorophyll minima is around 3.5 macromolar of oxygen, being between 2.1 and 21.3. The 90% of this chlorophyll minima. So this minima is very consistent. This minima, chlorophyll minima, in this very low light, oxygen conditions. So we think, or we hypothesize that the oxygen, or they are metabolisms that are dependent of oxygen, like photorespiration or water-water cycles. And these metabolism that present low affinities that could explain these patterns. And why this is so important? Because we know that these oxygen minimum zones are in expansion in the horizontal, but also in the vertical. So can reach also illuminated surface waters. And the question are, will it stimulate the presence of secondary chlorophyll maximum? Will it negatively affect the surface chlorophyll maximum? We really don't know how this can affect these biogeochemical cycles. So finally, some take-home messages. The AMC phytoplankton are photosynthesizing and producing oxygen, which stimulate aerobic processes. The secondary chlorophyll maximum alters the delta carbon 13 in the anoxic layer, and its absence increased chemoautotrophic activity. Although there is glucose assimilation in this secondary chlorophyll maximum community, it was not possible to corroborate that Prochlorococcus is using organic carbon sources. And temperature, oxygen, carbonate chemistry parameters have a significant influence in the distribution of phytoplankton, and oxygen-dependent metabolism pathway with low affinities for oxygen could explain this pattern in OMCs. This could result in significant inhibition of the water column photosynthesis by modifying biogeochemical cycles in the context of a OMC expansion. So this is it. Thank you very much.
Thank you, Montserrat, this great talk. And now we are opening the round of questions and answers. There is a first question here. It says, which data do you use to profile an oxygen resolution at temporal and spatial scales?
Okay, we use in the case of when we evaluate the profiles, we use the fluorometer in the CTD and also the oxygen sensor of the CTD, but also the stock sensor, that is a sensor that is very, very sensitive, reaching nanomolar scale. And there were several profiles in several cruises, and in every cruise we always found the same profile. Chlorophyll and oxygen reserve. And I don't know if I am.
Yes, I think so. I think we can move on to the next question. The next question is if this double chlorophyll A maximum is always present in the anoxic marine zones, or is there a seasonal behavior?
Yeah. No, it's always present. It's not a seasonal behavior, but it depends of the depth of the mixed layer, because sometimes when the mixed layer is very deep, there is not enough light to support this secondary chlorophyll maximum. Now, a couple weeks ago, there was a cruise in the north in Chile, and my colleagues they don't detect this secondary chlorophyll maximum because we also have El Niño. So there is not always present, but when it's present, it's always in anoxic conditions.
Good, thank you. Another question now from George Florin Staiku. He says, how the information offered by this presentation could be utilized by policymakers to stop oceans and seas pollution, ocean acidification, and surface warming? What would be the suggestions the policymakers should take into consideration?
Very interesting. First of all, we need to, we as a scientific community, we need to try to understand a little more of this. I think that we have a lot of questions because we don't know what is going to happen in the future. Maybe we can put all this data in models to try to make this data more easy to understand to the people that make decisions. And yes, it's part also of the is also part of the climate change. So it's maybe it's a point that we are not taking in account, and now we have to put in the in knowledge. Sorry.
Okay, Montserrat, thank you. Another question by Eleonora Puccinelli. She says if you have given some thoughts about effects on higher trophic levels relying on phytoplankton, for example, zooplankton or other trophic levels.
About the effects of, like, but I don't know if I understand what happened with the zooplankton in the OMC, or I think I'm trying to understand the question, sorry. What effects of the higher trophic levels relying on phytoplankton? Yeah, maybe we know that zooplankton also depends of the phytoplankton. So if these anoxic waters are affecting to this surface, phytoplankton maybe can be affected the zooplankton because they eat this phytoplankton. But so yes, I think so, but it will be interesting to evaluate how much can affect this zooplankton.
Okay. Montserrat, are you
See?
listening?
Yeah. Yes.
Okay. So we will take the last questions, but there are many others that are coming to the questions and answers box. So I would suggest you to continue the responses by using the chat or contacting directly the attendees. So I will take the last question, which is made by Augusto Franco. He asks, why did you use Imantonia to compare oxygenated and OMC conditions?
Yes, we use Imantonia because that first, it's a prokaryotic phytoplankton, and also because it was isolated from the same area. We try to use the organisms that we know that inhabit that habitat, and trying to see if this anoxic or low oxygen waters reach this area when we know that Imantonia inhabits. I don't know if I understand the question, but maybe we can discuss this later or write an email.
Okay, thank you very much, Montserrat, for your great talk. And well, I encourage you to respond the other questions that were left in the questions and answers box. And now we are moving to our next presenter. Montserrat, if you can stop sharing your screen. Yeah. Well, meanwhile, I will present Michelle Graco. Michelle is the leading scientist of the Chemical Oceanography and Geology Unit of the Instituto del Mar de Peru in Peru. She is also a professor of a Master Science program at Universidad of Lima, Peru. She develops research in the fields of marine biogeochemistry, coastal upwelling systems, and climate change with emphasis on carbon and nitrogen cycles, the oxygen minimum zone, and recently ocean acidification, particularly in the Humboldt ecosystem of Peru. Michelle coordinates different research projects with national grants and also participates in international collaborative research projects on coastal upwelling and OMC biogeochemistry, mainly with France and German partners. She takes part of the International Ocean Acidification Observation Network, GO2NE, as well as the Latin American Acidification Networks, LAOCA and PEMARCO. Finally, she leads the Peruvian National Report of the Sustainable Development Goal 14 Indicator of Marine Acidity, and is committed with several initiatives supported by international organizations in order to improve the research capacities for the coastal marine environment in the frame of the ocean. So, Michelle, you are welcome to this session and the floor is yours.
Thank you, Dimitri. Okay, well, good morning, good afternoon, good evening. Thank you very much for this opportunity to go on, and thank you for all the people that is connected. Today, I will try to present some results and initiative from the Peruvian system. We moved from the great job of Chile that present Montserrat, and now we are of Peru, part of the same upwelling Humboldt system. Okay, today during this talk, I will start with a brief review about the probably many of you don't know the principal characteristic of the upwelling system of Peru. Then I would like to look about the local effort to monitor the system and why it's important to monitor and obtain long-term observatories or monitoring in the coastal area. And finally, I will present some conclusion gaps, challenges, and opportunities for Peru and for the region. Okay, first of all, I want to say that, okay, the Peruvian upwelling system is a system that is driven by a physical process that is the upwelling. Here we have a cartoon. And the upwelling is a process related with the wind stress, the coastal area, and the rotation of the terrestrial rotation. And this made that coming from source sulfide waters with high quantity, with high content of nutrients, and this determined the high primary productivity in the system. This high primary productivity is dominated by a phytoplankton community, most of the case by diatoms, but in different conditions can be moved to a mixotrophic community, for example, dominated by dinoflagellates, and also epicplankton or nanoflagellates. And recently, we performed 2 studies of Peru in a control system in Mesocosmos, and we can test some hypotheses related to the upwelling and the impact associated with the nutrients content and the light intensity in the community. And then one of the things important and different to other systems is probably we don't have the biggest primary productivity, but the system is so efficient to have one of the bigger fish catch in the world. And this is the focus for many scientists, and that make many hypotheses associated with this condition. And what happened with this high productivity? Well, this is the upwelling system in the coastal area, but this is part of a more complex physical dynamics that in one part consider this high productivity and the high pelagic benthic coupling and carbon export that are related with the oxygen depletion, and also a complex circulation and the history of the water masses that made that on the Pacific we have the oldest water masses and one of the more intense and shallow oxygen minimum zone that we have here of Peru. Okay, the oxygen minimum zone is a combination of the high carbon export, low ventilation, all waters that have accumulative depletion, and this low oxygen condition is a characteristic of the system. And as in Chile, we observe that we can also have this anoxic nucleus in the oxygen minimum zone. And in another way, the high carbon export determine high remineralization of organic matter that determine another condition in the system, that is the high CO2 fluxes to the atmosphere. See, we have of Peru one of the main source of CO2 to the atmosphere, and the high CO2 in the waters determine also very low pH. This is presently work of Hernandez Ayón of Peru, low values of pH, and a very shallow saturation condition for aragonite and calcite that potentially affect the distribution and abundance of organs. And in another way, we have low oxygen, high CO2, low pH, and the processes associated also with the remineralization of organic matter determine a very important deficit of nutrients. It's a system that in the upwelling have a lot of nutrients that come into the surface, but in the same way, in the subsurface, we have a very important deficit of nitrogen. Okay, well, this slide is very crowded, but this is the idea, because probably we can observe different processes in a moment, but it's very important to know that this system have a very high variability. And here I can to boost different examples and publications. This is the result of more than 50 years of monitoring of Peru. And we can observe and recognize, for example, the seasonal variability. And here we can observe, for example, one characteristic that is that the maximum upwelling is out of phase of the maximum primary productivity. This is a characteristic typical from the Peruvian system. Then we can have also one of the most strong signals of the ENSO event. Now we are in El Niño condition. And here, for example, it's a time series of Callao that show that we can have different types of El Niño condition that make a different impact in the water column. Here, for example, we can look in different warm colors, the oxygenation impact of this El Niño event in the water column. And then we have decadal, multi-decadal trends, and in the climate change scenarios, we can also, we have some evidence of the expansion of the oxygen minimum zone from the next 50 years. Okay, high variability. And high variability in a temporal scale, but also in an spatial scale. No latitudinal changes, high mesoscale activity. And this is very important. Why? Because the challenge for us is try to sustain ocean observation in order to track this different variability and can understand what could be the future scenarios and how could be the impact for the commune. But what happened in the coastal area? Because many of years we are focused in the upwelling from the coastal to the ocean, and we explore a lot about the oxygen minimum zone, about the upwelling index productivity. But in the last years, we start to look more to the coastal area. The coastal area that is very important because we know, and this is a very interesting review of Larger et al. Of 2020, that we know that the coastal areas and bays offer a very high productivity conditions and are important ecosystem services that sustain economic and social activities. And this is very important because we start to look the coastal area, and a coastal area that is under the influence of upwelling, but also is under the influence of many anthropogenic and activities associated with the land-ocean connection. Here, one of the examples why in Peru is important to know and understand the system of the bay and the coastal areas. Here, this is a work of Mendo of 2016. Is associated with the natural banks of Arcopecten purpuratus, Ostión, or Concha abanico. And here you can say the export and the million of the export of these scallops, because it's a very high economic value. These artisanal fisheries and farming sustain thousands of fishermen, many people, and is one of the main artisanal fisheries of Peru. But what happened? Sometimes we also detect massive loss or mortality of these organisms in the coastal area. And the big question is, what produced this change? And to answer this, we only need to go to the data, the environmental data, and also try to have a baseline in order to compare if we have changes and what happened in the condition in this bay. And here I would like to present the work of several young people, early career students, and also senior scientists from IMARPE that developed a very nice work in Paracas Bay. Paracas Bay is in the south of Peru, is one of the places that was a lot of concha abanico in the past, and now it's a place that have a persistent hypoxic events during the year. The Paracas Bay is under the influence of the upwelling, but also under the influence of all industrial and different activities that are in the coastal, and also the influence of the river Pisco. When we look here, there is a implement a monitoring in order to track the changes. We can look for oxygen and pH in the bottom, that we have a very dynamic system with a high difference of oxygen condition and pH during spring, November, summer, March, and winter. And this kind of studies detect that a strong hypoxic event occur in summer associated with low pH, and this can affect dramatically to the aquaculture activity and the development of the concha abanico posture. And in fact, this is IGARSA under review result, but it's to show that if we have the monitoring and we have implementing this kind of environmental data, we can also then generate, for example, an hypoxia-biological effect index in order to understand what happened in this bay, what is the spatial variability, and what happened in time with this level of oxygen that can be, for example, lethal, sublethal, or innocuous for the organ. No? And here, for example, Mari Carmen Igarza characterized the hypoxic event and then convert this hypoxic event to a percentage of occurrence of condition that can be lethal or not for the concha abanico. And we can observe in the bay different spatial zones with different conditions, and this could be directly related for policymakers and to make a better management in the system. Okay, but what happened with the monitoring and the data and the around the world? And okay, I start to looking the different papers related with these coastal observatories, and I found this nice review from Selczynski of 2022. And well, this is something that is not a surprise, but is an evidence that in the north we have most of the observatories and the data available around the world. And in the south, we have very few coastal observatories implements. Probably we have a couple of more, some in Chile and in Argentina, but in general, the South Hemisphere is still, all the data is still scarce. And this is one of the things that we say, okay, but why? Why is this is the picture about the observatories? And for that, we decide to implement a project. This is a project that we started in 2021. This is a national effort of Concitec Prociencia. And the idea is try to implement a long-term, but another key aspect is low-cost environmental observatory in order to monitor all these environmental parameters or coastal stressor, environmental coastal stressor for organisms, principally associated with oxygen, pH, and acidification, and sulfur. And this is the team. This is a multidisciplinary team from IMARPE and also from universities, Científica del Sur, Federico Villarreal, and with a close cooperation with IRD and the Dr. David Point, that sustain also different undergraduate and master and thesis and postdoc projects in course. And also we are associated with an organization that is Pro Aquicultura, that make the link between the observatory and the fishery activity or the aquaculture activity. Okay, and this is the project that we start. And the other message is that we are not alone to implement this kind of observatory. That is a very challenge about the logistic, about the risk that we have to put equipment on the sea, and also to try to implement the innovation to have low-cost sensors in the system. And for that, we are in the frame of a big project in America Latina and the Caribe. This is a project with the international, the Agent of International Energy Atomic. And this project is important for us because support a lot of building capacity, equipment, training, intercomparison that sustain and also make more easy to have a long-term laboratory of Peru. Okay, the laboratory, this observatory is in Callao. This is the first. The idea is try to replicate and go to another latitude of Peru. We have a time series with more sophisticated sensor like CFOX, but also more low-cost sensor like Minidot and TPIT for temperature, for example. And also, we combine this time series station with a monitoring bi-monthly or monthly in order to complement this kind of data with other data like nutrients, chlorophyll, the carbonate system, the sulfur, and plankton and benthic communities in the area. Okay, and I would like to present two of the first results that we have from the observatory. One is the time series. This is our first time series, and in particularly for pH in the area. We deployed this system in July 2022, and it's very interesting because now we are in El Niño, and we have the opportunity with this observatory to complement other data and results that have Peru in the North area and discuss more about the influence and the impact in the area of El Niño coastal. Here you can see the temperature, the oxygen, the pH, and you can look that during El Niño costero, we have a very clear signature in this very coastal area. And this is good. And the other thing is we implement the master thesis of Wilson Carguapoma, and we start to report the index of acidity of the sea, and we start to put this data available for all the world. And this is almost the last slide. This is the other thing that we try to focus in this observatory, and is the innovation. We try to go to other kind of sensors that could be more cheaper, but with the high quality of the precision to measure. This is a sensor related directly with the importance in the system related with the sulfur plumes associated with low oxygen and high organic matter. And we use a passive sensor that we develop in the laboratory in order to track or to follow in the water column the conditions of the sulfur. This is very easy. It's a chip. It's a colorimetric reaction in a gel developed in the laboratory, and then we can transform this colorimetric reaction to a concentration and have the condition of sulfur in the sea. And this is part of the master thesis. of Kevin Diaz. And also, this is the platform for different undergraduate theses that try to look the microfouling and macro and microfouling effect that can have over this kind of sensor in the system. And okay, and the idea is to replicate that, try to go to other latitudes. This is the low cost is something that we can implement, and we don't need a lot of logistic. And the good thing is that we try also to make something very easy that not only the science can make the measurements and know the measurement, but also different stakeholders can be part of the monitoring to understand more about this condition. Okay, only some home message for all. The importance of the multidisciplinary approach to understand the complexity and feedbacks of these upwelling areas, monitoring as a key aspect that we need to articulate and integrate in a better way in this observing system. And this is very important to track, to determine trends and also the future scenarios for productivity, biodiversity, and ecology. What we need sustain this long-term observatory. We need good practices and improve the measurements in all the south part of the planet and in Latin America about pH, oxygen, the carbonate system. And we need also go for the innovation. We need to go to more projects in science to innovate, to develop low-cost sensor, not only passive, but also new technology and sensor to measure in situ, local calibration, reference material, and this possibility to monitoring with the stakeholders. And the last is we are not alone, and it's very important the networking and the international collaboration. This is a very a big opportunities for us, and I think I say to all the people that is a good match to stay in different networkings like Remarco or La Oca, GO2NE, and okay, and the idea is to have more observatories to know more about the coastal area and can have a better management of the biodiversity and the resource. And thank you very much.
Okay, thank you, Michelle, for your very interesting talk. Let me ask something first. Have you implemented an information system for the community? Is that in your plans?
Sorry, Dimitri, can you repeat?
If you have implemented an information system from this observatory for the community.
Now we are developing the, digamos, to implement the system. For the information, we only implement for the SDG, the Sustainable Development Goals. The information is open to the community and sent to the UNESCO. It's all about the pH and carbonate. And the idea is at the end of the project have open access information with all these time series. Yes.
Okay. Yeah, now we have here two questions in a row. The first one is how to participate in the project. What is it necessary? It's an anonymous questions. And the next one is, which could be the minimum necessary equipment to start?
Okay. We think, for example, the minimum in a system, in a coastal area, is temperature. And temperature is there are good low-cost sensors that you can implement. For us, for example, and in coastal area in general, the oxygen is also the other important sensor that you need to implement. Then the salinity, for example, well, if we have the opportunities, okay, but many times it's more complex. But if you start monitoring in the coastal area, you can implement sensors like tidbit or hobos with oxygen and temperature, and with that, you can track a very nice time series and understand about the change in the physical and the biogeochemistry. And then I think you need also probably to make a monitoring to complement this, this kind of high resolution information, and you can take some samples for oxygen, for pH. That is another important variable. But sensor in pH now are very, in general, is more the quality of, to measure a good pH value, you need more sophisticated sensors. The next step is try to look, what happened with the new pH sensor and compare for the quality of the CFOX or other sensor autonomy. pH is a more complex variable, but you can implement a very nice technique that is the spectrophotometric, and with this technique you can start, for example, a time series, and the precision is very interesting, and you can start to make in the future some trends associated with the pH.
Thank you, Michelle. There is another question by Rugesh Bhatti. He asks, the pH of coastal seawater was near about 7.5 in the time series that you showed. Have you seen any impact on biological organisms there, like on coccolithophores or other calcifiers?
Yes, well, thank you for the question. Yes, we have, well, there are some years we make a master thesis related with coccolithophores, and the coccolithophores in the upwelling cell, that is the low pH, 7.5, 7.6, we don't have coccolithophores. But then in low pH, but in still low pH, we can have coccolithophores, and we found a different morphotype with an overcalcification in the coccolithophore. And this is very interesting because the coccolithophore appear to have an adaptation or to this low pH condition, and we can found in the Peruvian system coccolithophore near the coast under low pH condition. And of course, in the oceanic part, more abundance and less calcifiers that near the coastal area. The coco, bueno, this is a very interesting research line that we expected to continue because there are a lot of questions associated with the, this kind of phytoplankton and the adaptation in the area. And for the Argopecten purpuratus of scallops, yes, there is also some studies, experimental studies. One important thing is if you know the condition of the pH in the system, this is something that Christian Vargas say a lot. Is we need to know what is the value of the pH in order to make the experiments and makes a real experiment with the organisms. And we compare, for example, the organisms in the low pH condition, but we go to a pH in the context of climate change, a lower pH, 7.4, 0.2. We start to look that the organisms have some impact. But it's different, the impact from an organism that is developed under an environment with this high acidity. These kind of organisms are more resistant but there is a limit, and we will, if you go to a more lower values, of course, you have change in the resistance, in the calcification, and also some compromise in the growth of the organisms. But yes,
okay, well,
probably the only thing is okay.
Yeah. We are running out of time. I'm sorry.
Okay,
yes.
Okay.
Yeah,
okay.
there is a lot of questions in the questions and answers box, and you could check it and maybe follow up the discussions with all the attendees. There are a lot of activity in this questions and answers box. So thank you, Michelle, again, and thanks to all the attendees. And now we are approaching to the closure of this session. Yes, so we look forward to welcoming you again for the next webinar in November. Also, please check out the Ocean Oxygen website, which is the go-to website for the ocean deoxygenation community. And with this, we are finishing. Thank you to all, and wishing you all a pleasant day or night from wherever you are connected. Thank you very much. Bye-bye.